with the rest of the team, it will help them in their revision and they can catch up with a few things that perhaps are not very obvious at one go. So pathogenicity, the other term we use is virulence or invasiveness and toxicity. Toxicity here denotes to toxin production that may be harmful to the host. Invasiveness is the capacity of the cells, of the bacterial cells or microorganisms in general to enter into various compartments. Usually, but microorganisms can attack the outermost cover, the skin, the mucous membrane. But when they get into blood, they can be transported to other sites, other compartments, which are very vital, and as a result may cause outright death in the host, or organ impairment, because those vital organs are critical in their functionality. We think of brain, we think of the liver, we think of kidney, and we equally think of the tissue structures. So if they are dismembered, if they are degraded, then the person suffers, the host suffers great disability. Not disability in the context of, the physical disability that you guys perhaps are familiar, but there are failure in physiological processes. So these physiological outcomes are very crucial for survival, exchange, coordination, metabolic processes, and of course, waste discharge. So if any fails, we have a big problem in that context. So microbial agents often would proceed and cause disease. So they have a very intimate, certain situations, they have a very intimate relations with the host, and certain microbial agents, microorganisms, are referred to as commensals or normal flora. So you'll hear terminologies that interchange in that way. So normal flora essentially describe the organisms that are found on the skin or on various sites and are not necessarily harmful. They promote even protection in what you call mutual beneficial interrelation between the host and the organism responsible. For instance, the notable part is the gut. We have so many organisms in that context. In the genitals, especially in the female genital tract, we have so many of these organisms which promote normal physiology, and they keep away other microorganisms with deleterious potential. And therefore, we focus on them. So we sometimes use terminology as commensals. And when you hear about commensals, these commensals may take advantage. And when they take advantage, we refer to them as opportunistic pathogens, because ordinarily they were not causing disease, but something must have gone wrong with the tissue or with the host. And part of that, therefore, make them take that opportunity and cause disease, invade the tissue even further. So the normal body flora are important, and we at present regard it as the last or one of the most component in context of structure, because there are so many microorganisms and which promote life for the host. We refer to them as microbiome, and which is a very big science today. And part of that is another organ altogether. It's the last organ that you may regard in far as the host is concerned. So the host microbe interaction, so sometimes may bring about distability, which it can be defined as violent defense reactions from the host. And some of these have got potential outcome. So one of the outcome outrightly will be that the parasite is killed and expelled completely if they become obnoxious, if they become harmful. Sorry, sometimes I may resort to using words that you're not familiar with, but if that occurs, you need to pick your dictionary and you follow it. So it would be obnoxious, relates to exposed to something that is unpleasant or harmful. So those organisms which are potential to cause harm will be then interacted with through the very varied host mechanism that will then remove it completely and expel it. So the host may be killed. That's another outcome. And in this situation, the microbe has got more powerful factors. And or perhaps it uses one strategy that creates disorganization in the host defense mechanism. So microbe and host therefore may sometimes also reach an equilibrium, sort of a balance, where they sort to live together and none harms the other for a time, because usually this is not permanent. And that time can be varied. It can be several years, it can be a shorter time. So majority of microorganisms will therefore cause diseases. So bacterial meningitis is one aspect that you guys perhaps must have interacted with even before you came to the university. And simply because it's a commonplace, you hear relatives say, he's suffering from meningitis. He's suffering from uh, pneumonia. He's suffering from boil. He's suffering from upper respiratory tract infection. He's got TB. He has renal failure. He has got sexually transmitted disease, and therefore highly in state that everybody else fear them and consider them very normally promiscuous in that context, in the context of sexual transmitted diseases, and which sometimes it's not the case. It's just as a result of what? Exposure to natural process, which is sex, and then pathogens can be transmitted. So some of the pathogens that can be transmitted as a result of sexual exposure could include chlamydia, trachomatis, Neisseria gonorrhea, Treponema pallidum, Ureaplasma urealyticum, Haemophilus ducreae. And around that, we have urinary tract pathogens, which are not necessarily sexually acquired, but sometimes sex may predispose to them, especially if the aspect of the genitals or sexual organs are contaminated with this pathogen. So we have Escherichia coli, Enterobacteriaceae that are other than Escherichia, Staphylococcus, Saprophyticus, Pseudomonas. Pseudomonas sometimes tend to be extremely very stubborn in the female genital tract and will cause deep-seated urinary tract irritation and dysuria. Dysuria is pain while one is passing urine. Very, very common in that context. And again, it's also resistant to most drugs. It's acquired resistance and can be a serious problem. In the context of food, we have Campylobacter jejuni, Salmonella, Shigella, Clostridium, Staphylococcus aureus, and Escherichia itself may promote disease, especially in children. Neonates are children who are just born. And these days, the word would go until, before it was up to 2 years, up to 2 weeks. But now they want to push it to 2 months. So they are referred to as neonates. And these neonates are very fragile in the context of exposure. So exposure could come from contaminated genital birth canals, genital tract, and which then could allow for contamination in the genital female tract, for that matter, during delivery. We have gastritis. Gastritis is the inflammation in the stomach. And Helicobacter pylori is a major responsible pathogen for some of these ulcerative disorders. We have, of course, upper respiratory tract, where we constantly interact with Streptococcus pyogenes, Haemophilus influenza, and obviously that can proceed to the upper part where we have sinusitis. So Streptococcus pneumoniae, Haemophilus influenza, and which can descend. They can come down and cause pneumonia, outright pneumonia, which is lung infection. And then we have Staphylococcus aureus, Neisseria gonorrhea, Chlamydia trachomatis, and so forth. So under the lung, we have again a huge group, other than certain viruses, which may also cause disease there. And in the brain, we have those that can evade, can go through, can permeate the meninges and cause disease. And as a result, therefore, their disease aspect is very, very remarkable, very powerful, and that can cause death and disability in the host. So infection and diseases that these organisms would cause would vary, and in the context of the host, therefore, the host must present itself with a formidable defense. And if this defense is not forthcoming, the tissues are at a great risk, and eventually the whole host may suffer. So these can lead to diseases which we refer to as apparent disease outcome, leading to injury, and of course, death can follow. In some situations, if the situation is formidable, as I mentioned, the host may then ward off the organisms. So what defines this disease aspect? At least the normal status would be easier to handle, because you just say, at least the immune system got rid of it. But what happens in this disease pathway? So along the pathway of the port of entry exists at different levels. So one of them is contact, which I talked about earlier. The other is inhalation. So you may acquire pathogens through inhalation. You can acquire it through contact, sex, and also some that can get into the skin can be contact. Then inhalation, those that you can breathe in through the contaminated air. You may also ingest them with food. And the fourth one was the aspect of congenital acquisition of diseases. So in the congenital context, the fetus or unborn baby is exposed to pathogens during the pregnancy, during gestation, and that can promote a lot of trouble. And as a result, therefore, their survival is in peril, is in danger, and they may either abort, the fetus may die, or there will be disturbances, like say, bleeding episodes, which occur many occasions, and that then require obstetrical care and adequate of it, for that matter, for the fetus to remain viable. So the attributes of parasites which may enable it to attack and injure the host include the pathogenicity, the virulence, the invasiveness, and toxicity. So pathogenicity would constitute several of these, sometimes together, or a certain factor individually. So virulence factor, one of the most important virulence factors is the capacity for microorganisms to replicate, to reproduce in large numbers. So in numbers they trust. So they become many, millions within a very short time. many, many millions within a very, very short time, and that overpowers the tissue. Secondly, they are they learn when to attack, which I called earlier quorum sensing. So when their numbers are not attained, they will not attack the tissue in any sense. But when the tissues are adequately served with so many of them, and they realize they can produce biological factors, then they will attack the host and promote serious degradation of the host tissues, and that then lead to disease. So they have that capacity to sense their environment, decide when to attack, and that is why certain diseases, you get exposed and you take several hours before they manifest. You take food contaminated with bacteria, but you don't get sick immediately. They start reproducing when their numbers are enough, then they express, they start protein synthesis aspect, which is gene expression to allow for pathogenicity. So some of that then can be very invasive. So if they started at a focal point, if they started at a local area, they will extend through various mechanisms. And sometimes they use the body's own transportation endeavors. And so they can be transported to very far distance sites from where they originally began the focus. And as a result, therefore, they arrive and impair. They arrive in vital tissues and impair those tissues, and creating disability, disorganization in the host. And that is the invasiveness. That is where they invade several compartments, several layers of the body, and become toxicity. So toxogenicity generally denotes the capacity of these pathogens to produce toxins. And these toxins can be very harmful, as you are aware. So we'll move on and look at them closely. So the pathogenicity therefore denotes the ability of microorganisms to cause disease, while virulence is the degree of pathogenicity, that capacity to cause in a tissue. So patho here is disease, and this disease manifests towards which sites. So the specific organ, and of course, what will happen to that organ. So non-pathogenic organisms, therefore, sometimes don't cause any sort of disease. So some strains of non-pathogenic organisms may have the capacity to cause disease, while others would not outrightly cause disease for that matter. So in that, those that do not cause disease, even if opportunity may favor them, are referred to as avirulent. Avirulence in biology, in virology, in microbiology, in biology generally, when a word a prefix is there before a word, then it means it's negated. When we talk about azoospermia, you've heard about a condition we refer to as azoospermia. It's a fellow who does not produce a male who does not produce sperms. If you have the other condition is agamaglobulinemia, someone who would not produce gamma proteins, and so on and so forth. Asexual reproduction. So asexual reproduction, it means sexual reproduction is lacking. So here, virulence is lacking, and so on and on. So virulence is a measure in terms of the number of microorganisms or toxins produced that put together, that collectively may kill the host. Sometimes the toxins may be there without the organism. In certain foods, I'm still not very sure. How many of you went to a party over the weekend? Or parties are under embargo? Anybody who ventured into some church activity and or mosque group, secular event that there's a lot of food served? Was there anyone lately, over the weekend for that matter? Nobody. And who ate from a cafeteria?
I did.
You did. Where did you go to?
I went for a birthday party.
Wow. And you ate?
Yes.
What was the food? Some rice?
It was mainly junk. It was like a whole, you could choose from anything. So there was pizza, there
Ah, the
was
buffet.
yeah.
I don't go to such places. If you go, don't eat. Just
Why?
take Desani and Coke if you want to survive better. Because if you do, you will eat toxins. Some of the toxins will come out from outrightly the people who are ahead of you. And like you mentioned, buffet is extremely dangerous because the people doing the serving, especially when the crowd is so large, they start very well. They serve very diligently. But at some point, they will get tired and walk away. And then the ravens decide to scoop for themselves. So sometimes the serving spoon is it's dipped inside and it disappears there. Or a small section is being seen. And the hungry lot, because at that point also the hunger level is very high. So the fellow says, no, let me just scoop it. And it's also not very hot at that time. So it will not burn, but there is some stew, some rice there. That is the recipe for diseases. So I myself, I never, if I go, I would ask them for more water and more sodas for that matter. Sodas would take me far. But you are lucky. You need to count your days. If you, when was it on Saturday?
Yes, Saturday.
So you'll have to count another 48 hours. So for toxins, they'll come after 48 hours because they're from that time you eat to 48 hours. Then you'll say you are safe from toxins. But then you start counting maybe another 48 hours for microorganisms because there is quorum sensing and then the productivity of factors. So some of those factors therefore will be microorganisms themselves or the toxins they produce. And these toxins can kill the host. Take, for example, we want to grow microorganisms to demonstrate that they can kill the hosts. So you can take, for that matter, a group of cells. Cells here can be blood cells, maybe white cells, or cells collected from urinary tissue. Sometimes we get cells in individuals who are not making it in life or through surgery. So if you removed tissue from surgery and grow the cells, you can add extract from bacteria, which have got no bacterial cells. I'm not sure whether you are getting me. So bacterial soluble factors, they are the toxins. Those toxins, if you add them into viable cells or viable tissue, you can see how rapid they degrade the tissue. And a small amount can destroy the whole city of Nairobi if you put it in water. So some of them are very resistant to chlorine and sometimes cooking or heating. So the number of microorganisms, therefore, which can kill, if they are more than 50, we call it lethal dose 50, if they are 50 or beyond. And this is a serious problem because it means half of the cells that are exposed to this can be destroyed through necrosis. And we perform that in the laboratory. I'm not sure whether I've made any significant different, rather description in trying to show you how you can demonstrate the 50% of cells dying. Anybody who had the aspect of this, please explain to us. Anyone? Hello?
Yes, the aspect of what?
Hello? Say it again.
No, I was just asking, could you rephrase the question?
Okay. So how would you demonstrate that bacteria is producing toxins? Very simple. So I say you can grow cells in a tissue culture vessel in a just, a bottle, sterile bottle. Put growth media and the tissue cells will grow. So these are cells maybe from the skin, from the respiratory and or genital tract, the lung, the brain. You can get such cells, especially sometimes from surgery. So if there will be surgery and you intend to get some cells for testing, so you can pick a bit of that, cut it into small pieces and peel off the cells so that the cells, it's a whole procedure. But what I want you to get is that if these cells are maintained in the lab, they are viable, growing in a lab culture vessel in growth medium, and the cells are growing happily. When you introduce an extract of toxin, those cells will be killed. But if they are killed only 10%, it's not a big issue, but still not good, very good. But if 50% of them die, that is a serious problem. And we refer to that as lethal dose 50. Lethal dose 50. So at 50% of the cells, very good. 100% death, which can be serious problem, is remarkable. So can know this every other time. Making sense?
Yeah.
Yes.
Yeah, so if you don't get it, please just say. I am happy that some people have said yes. But normally I'm interested in those who have not spoken, because I only had 3. Those who have not spoken, what did you get? I only had the ladies. Okay, since they are all quiet, I have nothing to do. Ian, Matiru.
Yes.
Ian, did you get it?
Yes, I did understand.
Great. Thank you, Ian. What about Cham Jura?
Yes, sir.
You did?
Yeah.
Timon?
I did.
Good. So I'll not bother anybody again. I think I'm too bothersome. So the invasiveness. So commonly bacteria cause disease by invading tissues. They multiply within them and spreading from the primary side to the logment side. And that is the from the basement. Okay. Base is where everything else comes, and the apex is where you see the outermost. Whether it is the genitals, there is the outermost. Where it is the respiratory tissue, there's the outermost, and there's the innermost. So cells will come from the basement tissue. We call it basal layer, and it will proceed and continue in that way. So part of this process, therefore, enables the cells to grow. So toxicity, is driven by toxins. So toxins are produced and as a result of metabolism. So the cells would grow and produce toxins. So when the toxins are secreted under protein synthesis, because they are proteins, they will then target their cognate receptors sometimes and enter into those cells and harm the cell. So this can be very deleterious outcome, very, very serious deleterious outcome. So the deleterious outcome here denotes cell death, which may occur as a result of membrane dissolution, cell membrane for that matter, or it may be focusing on chromosome digestion. It can focus on pathway inhibition and so forth. So bacteria then would produce several toxins and exotoxins. So endotoxin are part of what bacteria comes with. Gram positive, or rather gram negative, have got this. Gram positive also have got a number of endotoxins. We talked about, in gram positive, we talked about teichoic acid. And sorry about that. Sorry, somebody has invaded me here. So sorry about that. Someone just ventured into an area where they are not required. So the endotoxins are factors that are generated but are held within the cell membrane, whereas exotoxin are factors that are secreted. So like I mentioned, the lipotechoic acid and techoic acid are part of the cell membrane, cell envelope. In the bacteria, we refer to as gram positive, and they can cause a lot of problems. They can activate the system and harm the system in that process. Exotoxins are commonly produced by gram negative, and they're secreted outside. Small doses, they are extremely fatal in small doses and may have a lot of harm in the context of pathogenicity. So various symptoms would then emerge, especially they may cause lesions of various types. Lesions are cells peeling off at a point, at a focal point. That can be in the brain, can be anywhere. And look at that. That can be extremely harmful. So endotoxins, as I mentioned, are structural. They are part of the bacteria. We have lipopolysaccharides, polysaccharides, and glycoproteins, and usually form part of the cell wall. Some can be found in gram negative and equally gram negative, gram positive. In gram negative, we have some we refer to as autolysins. Autolysins cause self-destruction on the bacteria autolysis, but can also harm the host very firmly. We have a portion of this can be entered into the system, especially the lipophilic area, and be delivered inside the cells. And so when they enter into the cell, they stop protein synthesis and repair, and as a result can be a lot harmful. So when I talked to my sister there of the risk she had exposed herself to, only to venture into a party and get some food, is that some of these toxins are thermostable. So if you boil them at 100 degrees centigrade for an hour, they would still be having capacity to cause disease, and which are themselves very harmful. And so some may remain at 60 degrees for 1 hour or 20 minutes. So sometimes foods that you eat and cooked or semi-heated or heated to some degree may have some challenge. But I have, again, encouragement that you can take a lot of water sometimes to dilute them, because they act within the gastrointestinal tract. So if you put a lot of water, we'll wash them off so they don't bind, but generally can cause a lot of trouble. So people die, people suffer when they eat from a common pool, a common scenario. So infection.
Excuse me, sir.
Yes, dear. Go ahead.
Will you go to the previous slide?
No problem.
Here, the fourth sentence. What do you mean by
The fourth bullet.
Yeah,
The fourth
this.
bullet. Here. So all endotoxins are emetic, that they would make you vomit. They will make you vomit. So when you take them, they act on the gut and make the gut peristalsis be either reverse peristalsis, and it also interferes with the stomach bile content and muscle movement. And the stomach wants to expel anything around there. And they are also pyrogenic, they cause fever. So when you have food poisoning, fever sets in very quickly. So majority of times people make a mistake that they take antibacteria. Is this going to be useful? Anybody, do you think antibacteria will work?
No.
Anyone? So emetic is something that causes vomiting. Antiemetic is the one that prevents vomiting. So tell me, why do you think it will not be good to use the antibacteria? So okay, first of all, my dictionary says that an emetic is an agent that induces vomiting. So you got it correctly.
Yes.
And the pyrogenic is fever causing. So it will get in, so they can do the two at the same time. And they are also necrotic. They also cause floughing of the cells. So when you go into the gastrointestinal tract, it's peeling off, just coming off completely. And that can be a serious problem. Then this also causes, through that, diarrhea ensues. They also cause what? Diarrhea. Diarrhea is the rapid movement, and so sometimes you can expel food before they are even digested. It might be beneficial for these toxins, but sometimes they can be problems. So for the endotoxin, you can use antibiotics because they are on the surface of the bacteria. But for exotoxin, that can be sometimes a problem because antibacteria don't act on it. Antibacteria will kill viable bacteria. But remember, this is produced in food, but the bacteria is not even there. But these can remain stable in heating. I'm not sure whether I've made a difference in that context. Did it make a difference? Or is it clearer?
Would you explain again why antibacterial doesn't work?
Okay. So antibacteria is targeting bacteria that is able to replicate. But since we say endotoxins are part of the bacterial cell, that could be helpful because you want to destroy that bacterial cell. Although sometimes that can also be dangerous, that you release those endotoxins into the circulation. But that can be dealt with at a secondary level. So outrightly, you don't always just give antibiotic. Especially sometimes it's exotoxin. So you make food like rice, you left the food overnight, but it is contaminated with bacteria. Bacteria may produce exotoxins in it, but the bacteria may be killed if you warm it, but the exotoxin will remain viable. Antibacteria does not kill exotoxin because these are just fluids, these are just solutions, while bacteria is a cell. So is that making sense?
Yeah.
Good. So we'll also talk about them when we perhaps come across a strategy in treatment. So terminologies, therefore, that can be used to refer to certain aspect of these interactions can be varied, very, very varied, and can be interchangeably correct. So relationship between host and microbes, we denote what you call a commensal. We denote by word commensal. So commensal are microbial agents that receive benefit, but there is no harm to the host. They receive benefit, but there's no harm. Sometimes they may also help the host to some degree, but not always. Opportunistic microbes are those that receive benefit and is able to cause diseases if given opportunity. And pathogenicity, therefore, is the ability of an organism to cause disease. Wherever they are there, they will cause harmful outcome. So ability therefore for these pathogens to thrive vary according to many things. One of the things I've mentioned is their colonization. They are many in numbers. They can produce factors, and those factors are what we are concerned with. So therefore, if that is the case, the non-pathogenic organisms have no factors, have no genes for disease. Opportunistic organisms are like pathogens, but their genes are not expressed at that. And they sometimes may also acquire genes and what you call a mutation, alteration, and so on. So transmission is the movement from one location to another. It can be on the hand to the gastrointestinal tract. And this then enhance the organism numbers wherever they are. And they can be large in numbers for that matter. We also may enhance their availability to survive extreme conditions. For instance, certain foods we eat may promote microbial survival. Like if you are eating seafood is usually salty, and then you also increase the salt concentration in the gut, especially by putting so much basic components. That outrightly could promote the survival of the organisms that love the salinity, and they therefore can cause disease. So some of these could be promoted. I am referring to Vibrio cholera. So Vibrio cholera is a disease that is acquired from seafoods. It's seafoods, please mark my word. They are normally not found in freshwater marine organisms, but commonly in the sea, because they love alkalinity of even up to 8.6, of pH even up to 8.6. So if you increase the pH in the stomach, the pH in the stomach is usually 2 to 4.6. So if you increase it to 8, then you make those organisms survive. And that is why in certain situations when people eat certain foods from the sea, they put a lot of lemon. Lemon has a lot of acidity. So that discourages any establishment. I'm not sure whether they know why they're adding it, but sometimes it's practice that has been carried over many generations, and why they don't understand, but some insight, intuitiveness give them the opportunity to defend themselves out of that. But perhaps they don't realize that whatever they are doing is a great benefit. So infectivity is the ability to penetrate a healthy host's first line of defense, which we refer to as innate. And this then can promote colonization. So colonization would then be followed by virulence. So they would be many, and they can express their virulent genes. And this enhance infectivity, enhance the disease process. So pathogenesis is the process that covers initiation of an infection process from the beginning to the point where you start feeling the symptoms. So as a result of this, therefore, certain outcomes may be obvious. One of them, lack of symptoms, may be referred to as asymptomatic. So when you don't show any symptoms at all, it's referred to as asymptomatic. And I'm referring to biological description: asymptomatic, azoospermia, asexual. And the other one we talked about lately there was what? Avirulent. So there is no symptoms at all, but there is infection. So if you engage laboratory testing, you'll find that there is there are presence of microorganisms, and may be unnoticed but detectable in the laboratory. That is, if we take a stool sample, we can test it and find that somebody has cholera, has Vibrio cholera in their system. Majority of this is the reason why we perform evaluation of the food handlers. So if from time to time anybody going to handle food, you ask them to undergo tests. I'm not very sure whether that is followed keenly, but we have unscrupulous officers who in the public health who would sometimes just issue certificates and close their eyes and fold their palm, going home very happy because something has been put in the palm. They call it greasing of the palm, and that can be a problem. So visible symptoms like a swelling at the site of entry or localized lesions or any of those can denote a disease process. And factors like sore throat, diarrhea, and so forth will be symptoms. So those are some of terminology that you interact with. And the virulence, therefore, is the capacity to cause damage to the cells, to the tissue, to the organ, and to the organism itself. And can be described as a virulent or cannot cause disease, or virulent, having potential to cause harm, and very virulent, now we qualify the noun here, or extremely dangerous in context of disease causation. So factors that relate to the virulence will include bacterial factors, so ability to initiate an infection. It may also include the presence and production of structures called bacterial ailment structures. This can be including those biological factors like enzymes and toxins. The other aspect is the adherence. So bacteria would use various structures to attach onto each other and to hold onto each other and to attach onto the surfaces of their hosts, on their hosts or in the organs. And so with that, they can create problems by being many. I said one of the factors that promote disease outcome is colonization. The other aspect is the aggressive factors like that include formation of capsule. Who can remind us what capsule was doing from our previous lecture? Anybody?
Does it help with attachment because it has like a mucousy layer?
Yes, so
Attachment
that
to
will
other.
help them in attachment, and it will also get rid of. It will also get rid of immune defense like the phagocytosis. So it prevents phagocytosis, antibody activity, and so forth. So it is important. So phagocytosis will not be there. It also, or some bacteria may produce enzymes and enzyme-related factor. Others, a mass high concentration of ions, like say phosphorus, iron itself, potassium, magnesium, and so on, but in a unique form. This, when released, can be harmful to the cell membrane of the host, especially those that live intracellularly. And we refer to this condition as siderophores. So those ions can be stained, and we refer to them as siderophore. Then we have the plasmids as the capacity to acquire genes, external region, and bacterial toxins, which I've talked about. So virulence factors, therefore, must have a property to cause disease in the host and may enable an organism, therefore, to evade host defenses, may improve access to body's nutrients, colonization, which is defined by attachment, and of course, promoting survival, preventing itself from antiphagocytic processes, which include capsule formation. So they will form capsule to prevent phagocytosis, to defend against the host, They will also have capacity to colonize. Colonization is defined by having capacity to attach to each other and grow in mass, in large numbers. They, while having grown, they will produce exotoxins, which are secreted in disease, and so on. Of ways can be in certain bacteria, they all might be available. Others just have specific what I mean, what I described earlier as having able to be converted by use of plasmids. So acquisition of plasmids may be a factor that is not very common to all bacteria. So substances that help organisms to invade would include hemolysins. I want you to mark groups like streptococcus. Streptococcus produce a lot of hemolysins, streptococcus. They also produce a lot of leucocidins. So hemolysins generally will damage blood, not necessarily blood, red blood cells, but red blood cells is easier to demonstrate when lysed, but they can damage all other cells of the body, even white cells. White cells are critical in the context of defense. And so when they are damaged, that can be a serious factor. And I think some of these factors, in our next lecture, I will spend time and show you a video, or I will upload a video that can be promoting that degradation. So I'll look for a video. There are many, which denote virulence strategy. The other aspect, which is again for us, the factors like leucocidins. Leuco means leukocytes, but they can be degraded. Leukocytes form the shades of groups that can promote killing of microorganisms, and they are the white cells, leukocytes. So if you kill them, then there's a great free for all, and the host suffer. We have hyaluronidase. Hyaluronic acid is a component of structural, connective tissue, and any other. So if you can have an enzyme that removes the structures, then that is a big problem. If it's in the gum, your teeth falls off, just drops into your tongue there, and you wonder, why is this teeth coming by? Can be a serious problem. Collagenase. Collagen is again factor that we find maybe in the joints, and it can be degraded, broken down. That is a serious problem. So toxins are part of disease causative agents, and they, as I mentioned earlier, we have enterotoxins, which attack the gastrointestinal tract, but usually from the stomach, the small intestine, and large intestine. They lead to diarrhea. We have neurotoxins, which inhibit normal neurological dysfunction. We have protein synthesis, which inhibitors and can kill cells, damage them profoundly. And then we have superantigen. So superantigen generally are activating the immune system, where the immune system become over agitated, over active state, and which for that matter leads to confusion, and you may go to toxic shock syndrome, toxicity, which is leading to shock, and that is complex. Staphylococcus, we are going to start on them perhaps even shortly. If this ends early enough, then we'll start on it. That can lead to damage, that can lead to specific damage. We have superantigen, and these are toxins that bind to microorganisms and then promote recirculation. The protein synthesis inhibitors, they stop formation of proteins, and so there can be no repair. Organisms like diphtheria would perform just that. And the flesh eating is another group we refer to as streptococcus. They can eat your flesh until you remain with the bone, maybe the arm. They start on the skin, and shortly you wake up the next day that the bone is exposed, and you're not even feeling pain. Tissue is just degrading itself, and shortly you are gone. Others may form gas. You have gas formation capacity in the tissue, and when that gas bubbles off, the tissue remains bare. That can be a serious Clostridium can do that, and several other mechanisms. So with all this, we have to be aware. So endotoxins generally are pyrogen. They cause fever, found in the outer part of the organism, and mainly gram-negative organisms would have them. They cause fever, and they may also lead to drop in blood pressure, leading to shock, and acts by binding macrophages and causing the release of substances like cytokines. So you go into cytokine storm. Cytokines are small molecules produced by the immune system or the cells of the body to coordinate. They are second to hormones. They are second to hormones. And as such, therefore, very important in diseases. So infection generally, in summary, therefore, basically means a process in which microorganisms invade the host and lead to harmful outcome, lead to an harmful outcome. This harm may then relate to tissue degradation, organ failure, necrosis, and perhaps the host death. So reservoirs are sources of organisms that could be found within the host itself or within other animals. Like say, we live with rats around. They may be reservoir for a specific pathogen. We live with cats, dogs, and several other, cockroaches. They can be a reservoir where organisms replicate, but when they get into our food, it becomes a problem. So human infection can be exogenous or endogenous. So exogenous means when you eat food and that food is contaminated. It can be from the environment, can be from water, can be any other source, or from another person themselves. So if you touch them, you may transfer it into your system. But ordinarily, certain pathogens arise as endogeneity, whereby the host harbors some of these organisms, and when the systems fail, opportunism is undertaken by the pathogens that now become outright disease causing, and that can lead to disease formation, and which can be a serious problem. So diseases therefore can be transmitted through various ways. We have respiratory tract organisms that include those that will cause secretions to form in the respiratory tissue, causing aspect of, or as a similar situation like drowning, whereby you can't breathe properly. You feel like you are sinking in, immersed in a fluid. And that can be a serious distress. They cause death if not watched. We have skin and wound infection that is coming into contact. So in the first one, we acquire them through inhalation. You inhale them, and also through contact. They can transmit, they can travel through blood from skin to establish in the lung. Some of the situations, other infections like say parasitic, may deliver them to the lung, and they can cause serious disease in that case. Pneumonia like outcome. So skin and wound infection are a major aspect that we must be aware of. So certain situations may also proceed, and these are defined here as a sexually transmitted pathogen that you only acquire when the genitalia get into contact. for purpose of reproduction. Although these days that might not be, is for fun and can be disastrous. You guys have heard about sex parties? I know you are adults, so there's nothing embarrassing around here. You've heard about them.
Yes.
Great. So those are some of the serious exposures vehicle. So you imagine one person coming there with gonorrhea and they want to have sex with everybody in the party, maybe party of 20 people. So that can be a serious problem, leaving each and every mucosal tissue exposed to the pathogen. It's not one of the best. So the outcomes have been disastrous. So transmission, therefore, you look at the source, the load, and the type of organism. The source, the load, and the type of organism. So sources can be food, sources can be another animal, sources can be our own system, and so forth. And other sources can be those animals we interact with, family, by maybe via food, environment, or vector. Vector, like malaria-causing pathogens, would come and bite and deliver the parasites. And this can be a serious outcome. But the format are factors like inanimate objects, like those that you use for slaughtering, for skinning, or for cutting. And sometimes they may cut our skin. Or sometimes food remains from what you outrightly share. You go to the hospital and this person is sick, but you are compassionate, you are empathetic, and you are showing love. So you share their food. That can be a serious problem and may give you trouble. And that then defines our discussion. So after that point, if there are anyone who feels they want to change their course and go back to do history, is the calling still there or the calling is dying away? You thought you were only going to talk about computer programs, and somebody has bombarded you here with a lot of words in medicine. Are you happy or you are bewildered?
Yeah, okay.
It's okay.
It's okay.
Good.
Interesting.
After that point, we can let that rest, but you read it. I was doing this. I normally don't teach it to other groups. I made it specifically for you so that when we talking about the pathogens, you have some idea. And I'm happy to say that I've loaded most of your notes in bacteriology. I think I've completed. Whatever is remaining is parasitology, mycology, and virology and immunology. So those ones will be covered in a record, I think 5 weeks. So I have the notes, but I couldn't put them because I'm trying to balance planning family to see which one is going to be better. And then you didn't tell me which day is for is practical, because I want us to start practical. The class rep. You didn't talk among yourselves.
Yeah, I'm so sorry. It
No,
skipped me.
don't worry. You don't
But
have to.
yeah,
You don't
but
have to.
let me say by the next lesson,
Yes.
I'll come with it. We'll discuss on the WhatsApp group, and
Yeah,
then
okay.
I'll initiate the discussion. People will suggest their days, and then I'll get back to you as soon as possible. Sorry about
I'm
that.
also part of the WhatsApp, so we'll see. Or you have another WhatsApp for
No,
putting things.
no. It's the same WhatsApp group. You want us to discuss in a WhatsApp group that you're not part of, or?
No. It's okay because there's nothing you can discuss which is out of the context.
All right.
Yeah,
By the next lesson,
we are normally only worried that they'll say it's too floppy, he does not know anything, and he's reading for us the slides. But which I know is a part of life, so you need to voice things out. But you can just talk. That is the I'll be watching from a distance. Then I interact wherever is necessary. So did I stop sharing for the earlier ones? Are you seeing Staphylococcus aureus? I would rather Staphylococcus and Micrococcus.
Yes.
Good, good. So that this will define our next discussion. And now we've started with organisms, diseases. But with that background, it will be helpful. There's a book that I need to give you, and my WhatsApp. By the way, for your, how do you deal with WhatsApp? Just teach me 1 minute to show me how it's dealt with. Do you normally delete them on arrival?
Delete the messages or?
The messages to allow the phone to be functional.
Oh, I think you just need to clear the cake, like the, I don't know how it's pronounced. Is it cake, cache? You just have to clear it so the phone gets more space. But
Like when you go to your settings,
Yes.
you can find your device memory.
Okay.
And with your device memory, you can be able to sort out what you need and what you don't need.
Because I have a very good phone, Nokia 6.1, which was working very well, but now I can't download any PDF, anything, because I think it's full. And you know, I'm in almost, I would say 20 groups, social, some scientific, some from school, some from, so many. And I think I just need to delete them one day and start afresh and tell the people to invite me afresh. But that would be challenging. So I'll follow your advice if I can deal with it. Yes,
Sorry.
go ahead.
Also, media sometimes automatically downloads. So if they send videos or pictures, it could download onto your phone, which takes more space. So I
Oh,
think just check your gallery for like WhatsApp videos or images and delete the ones you don't want.
okay. Thank you. I think that's what I'll need. I think I'll just spend a weekend seated under a tree with a coke and do that until everything is gone. So I had the book, but I can't access it. I can't download it to send it to you. So what I will do instead, I'll give you the name of the book and the author. So sometimes you guys might get it better than myself because it's getting late. Of course, we are still working on, especially from today's lecture. You still got there's one I also asked you to read, which was from the library. You remember Brock Biology? Or you also forgot about that? I see.
So
Sir,
Yes.
we went to the library and the librarian asked you to communicate because there was an issue with the book apparently.
That specific book?
yes.
Yes, it's under microbiology 2361. So they need your
There's nothing like that. We don't have 2361 on session. I know that is the situation. So I'll have to write to them. Didn't communicate back,
Oh no, I was going to when the class ending.
Okay, so please let's be proactive. That you just put immediately in the wall, because I'm seeing everything you guys talk about. And I see everything. So I could have responded by that time. But I'll respond to them. I tell them that it's also part of 2360, which you need. Actually, you don't need it from today. You needed it up to where we have reached. So you are having a backlog. So you'll have to go and get it. The other one I was talking of is what we now need from today, from this lecture. So Staphylococcus and Micrococcus tend to be one organism, but there are slight differences. The one that causes a lot of diseases are Staphylococcus. And now bacteriology begins. I was preparing you for the ground. Bacteriology begins in the context of disease process, and which I don't want you to forget. You will use this information throughout your life, especially in the lifelong career choices you have made to disease tracker and disease controller. Your main work will be tracking diseases and controlling them. Of course, you are working with the team, and the team can't work without you. So you are part of the team, so as myself and other experts. So we are all in this together. So this information I'm giving you is going to create stimulation into your understanding. And so in the future, you seek information in similar way, and you advance in that. So we have these organisms divided into gram grouping. So they are gram positive. They are all gram positive. So Staphylococcus are gram positive. The other capacity that they show is that they are catalase positive. So we have the group that can be divided into coagulase positive and coagulase negative. And that will give you a Streptococcus epidermidis and Saprophyticus, which are novobiacin resistant or novobiacin sensitive. Up to this point, I know I have created fear in you a great deal, that you are not here to perform what you call cramming. But it's simple. These organisms, they are gram positive. And as word would go, in the micrococcus, they ferment glucose through oxidative strategy, and the other would use catalase as a reaction. So those that use catalase, there are the Streptococcus and Staphylococcus. We are going to start with the catalase positive. These catalase, I know you're wondering now, what the hell again? So the catalase, the coagulase are just a factor of gene expression because they are enzymes. So the organism will produce an enzyme, which we refer to as catalase. So if it's producing catalase, we refer to them as catalase positive. If they don't, we refer using coagulase, they'll be coagulase positive. If not, coagulase negative, and so on and so forth. So those are not things that you cram. They come out ordinarily. So Staphylococcus is a Greek word which is derived from formation, the potential to form staphyle bunches. So Greek here was describing, a Greek word here they were using to describe the nature, the shapes, the pattern, and which they equated to grapes. When you look at them, they look like that outrightly. And they are non-spore formers. They don't have spores. They are non-motile, never move. And some are capsulated. When we say some are capsulated, what comes to your mind, my good guys? I've talked about capsule many, many times. Okay. Yeah, go ahead, sir.
When they're capsulated, they have a cell wall.
No, not really. When they are capsulated, they have a capsule.
Oh.
And that capsule was preventing phagocytosis. And phagocytosis is destructive. So that is the main thing around it. So therefore, we have the factors in these that we need to interact with. So some of those factors, therefore, their capacity to grow. So they will grow in salt concentration up to 10%. Most organisms may not tolerate that because the salts will interfere with their cell wall, cell membrane, cell wall, and all that in that context. They are also catalase positive. That means they produce catalase enzyme. And this enzyme if you have the enzyme catalase, it can break down hydrogen peroxide into water and oxygen. In a very brief moment, very rapidly, you have seen effervescence. Effervescence is bubbling that you can sometimes may observe in systems. So staphylococci are facultative anaerobes. They can grow in the presence of air or not. And in this respiration, therefore, they will form lactic acid. If lactic acid is forming in your muscles, what do you think will happen? Guys of physiology here, what will happen? Who among you is a long distance runner, a marathonian? Anybody? But who has gone jogging for considerable longer distance? Not very long. Don't look at it as Kipchoge Keino. It's not here. But as any of...
So
Yes, go ahead, sir. Go ahead.
I think if lactic acid forms, you'll get muscle cramps.
Yeah, muscle cramps. So this can form anywhere, even when you are seated. It will produce a lot of this lactic acid. You get so tired, pain, and the muscle cramp is just a statement. It may cause, where there are higher concentration of lactic acid, the cells die. The cells will die. So at least 40 species have been described, and most of them are generally harmless because they don't have the capacity to cause disease, but they live on the skin as commensals. So they live in mucous membrane, and they also will be found in nature, in the soil, where they perform putrefaction. But outrightly, they will harm the tissue in certain quarters, certain processes. So these are the bunching, the bunches that we refer to as the staphyle in Greek. And so the positivity, I think you have to remember. Before we go any further, let me hear from my good students. Who among you can remember the nature of gram reaction? What was defining gram positivity and gram negativity from this class? And very, very quickly, as I always say.
Should we talk about the staining or?
Is the staining. And so what about the staining?
Gram positive stains more, I think, a darker color.
Yeah.
And negative less stained. It becomes less stained, or the opposite, I don't know. But I think
but what was the principle? What was governing it? I talked about it very firmly. I talked about the thickness in peptidoglycan. So gram positive have got a higher thick peptidoglycan. Gram negative have got a thinner peptidoglycan. And so when you treat it with what I call remover for the ladies, nail polish, I guess, remover, polish remover, acetone, it will clear off, leaving nothing, leaving void in gram negative. So when you apply the second stain, it will absorb the gram, the second stain in gram positive, in the gram negative rather. But in gram positive, the cell wall is so large that even if you remove the acetone, it will only remove a bit under a considered time. If you leave it for a longer time, obviously everything will be dissolved. But if you remove it rapidly, like you put it and then remove it, it will only go with a section. And when you apply the second stain will not get anywhere to bind to. And that defines the positivity and negativity. Is that clear? Because
Yes.
now I want you to apply all that knowledge we started with in first class to today. We are building onto it. So ideally here I should have just said gram positive and gram negative and I move. But I know I appreciate where you are coming from and you have not interacting with this for so long. So I have to be very, very flexible and going back and forth, back and forth. And that is when you can achieve, or I can achieve my goal of having you guys well acquainted with the microbiological processes. So in clinical practice, therefore, we can group them into coagulase positive and coagulase negative. So coagulase positive are more harmful. And one specific group is referred to as Staphylococcus aureus. Staphylococcus aureus. Up to that point, is it okay? Where would coagulase come from? Coagulase come from the genes of the organism. So it has the genes that is capable of producing coagulase. Coagulase negative, they lack those genes, so they can't produce coagulase, and therefore they are not very harmful. Coagulase, as we had said just very briefly, will cause clotting. Clotting. Those clotting phenomenon when blood is clotted is a bad thing. If it clots in your veins, in your vasculature, in the heart, in the tissue, will be a big problem because you die of asphyxia. Whereas in the coagulase negative, they lack that potential. So even if the system, they will never cause harm. So they may be pathogens, but using a different strategy, coagulase. So we have the coagulase negative and coagulase positive, and in which the Staphylococcus aureus group are the ones that are referred to as Staphylococcus, or rather the coagulase producing, because they have the genes. And coagulase negative are many. We have the Staphylococcus epidermidis, used to be called Staphylococcus albus before. It used to live in the sebum, secretion in the skin. It still does. The name was changed to epidermidis because it lives in the epidermidis. So Staphylococcus saprophyticus is found in vegetation, degrading vegetation, and creating trouble for that matter. And then we have Staphylococcus hemolyticus, Staphylococcus lagdunensis, Staphylococcus schleiferi. So these are names that you need to start interacting with. Not cramming, but it will become obvious. Not cramming, but it will be very, very obvious as we move along. So the process, therefore, will require that we look at their characteristics and we can demonstrate them in the lab. That is the essence. That is the main goal. We can tell the difference between Staphylococcus aureus and the other group. So the Staphylococcus aureus, therefore, is facultative anaerobe. What did we say? What did I say very shortly that facultative anaerobes would denote? Just when I was beginning my lecture, I talked about other, not all in the middle. Anyone who can remember?
Does it mean that it can grow in the presence of oxygen or without oxygen?
Thank you. Yes, that is the nature. They can grow in the tissue, in the lung, or anywhere else because they don't, air will not harm them. They can have a wide range of tissues they can grow in. Commonly, organisms that are facultative anaerobes cause disease because in the tissue there's very low level of oxygen. And so if they can survive, it's good for them. So as we move on, you will realize, therefore, that majority of them are part of normal flora on the skin and on the nasal passages. So here, if you look, you collected material from pustules, pustular exudate, if you have boil, and many of you have suffered boils. And if I am not very wrong, sometimes I am wrong and people correct me, nearly every person here suffered boil. It starts with a small pimple, then it enlarges. You know, certain ages are more prone to boil. And if you've not, then something is not very right, and not normal human being, because these organisms are there and they are taking root. So it starts getting limited as we grow older because of the immune pressure that we exert. But if that immunity will go, these are some of the things that will kill you. So on the skin, therefore, you'll find many of these in the exudate, that pus that comes from your deceased buttock when it's producing, when it's undergoing boil process. If you collected a lot of that exudate from the buttock, I don't know why it loves that area, but there's a reason. Or in the skin, other than the buttock skin, it can be on the face, eyelids, the cheeks. But when you extract that, you end up with, and you stain it through gram staining, you end up with this. So I want you to know how you can perform gram staining. I talked about it in class. Family, if you don't know by today, you have a problem. Make sure you go back and know how to perform gram staining. And they are the two things I'm going to put video on. I'm noting gram staining. The second one will be disease aspects, diseases, how they may present. Atlases that could be available in the YouTube. So that will be useful. So these exudates, you make gram stain, you'll see this. So this is a mass of tissue cells, but these are the bacteria. Those tiny things are the bacteria, causing serious replication. So the capsule, this is a pathogen. This is how it is now. We have the cell membrane, we have the peptidoglycan, and then we have a capsule. When they have a capsule, they are more pathogenic. It's good we've talked about the pathogenicity, now you understand. And the peptidoglycan here cover it. That peptidoglycan is what will define the disease process. And then we have phospholipids, we have the transporter proteins, and so on, which are in the plasma membrane. So we have teichoic acid, which are also part of the organism, and lipoteichoic. So part of the teichoic acid will anchor various layers onto the organism. And then we have lipoteichoic acid, which anchors the teichoic acid onto the cell membrane, and that promotes survival. One, I don't want to go beyond your time, and I can see it's beyond time. So others, we have protein A. This protein A is supposed to be immunogenic, but it's also found within the peptidoglycan. Apart from that, then, the capsule is factor of pathogenicity. The capsule is a factor in pathogenicity. It's antigenic. It will promote antigen responses. It will also promote antiphagocytic. So phagocytes will not come and pick the whole organism. Secondly, now what I'm showing here, guys, is just a cross-section of the organism. It's a rounded material in that nature with the nucleolus inside here. We have talked about the bacterial cell. So I want you to relate to that cell, but we are only focusing on a small section encompassing the cell membrane, the peptidoglycan, and the capsule. And further, just allow me one, we have the binding protein, the phosphobinding proteins, and so forth and so on. Some of these, therefore, can be teichoic acid, which binds to the fibrinectins on the surface of the host, protein A, which has great affinity for Fc portion, and immunoglobulin, making them not be able to function. And this is very interesting. These can also cause what you call toxicity, toxin that lead to shock, and will continue from there. Any question? Any question? What are you going for, guys? After this?
calculus.
Oh, okay.
But lunch first lunch.
Yeah.
Oh, you guys are still able to do a little lunch.
Mm.
That is luxury. You know, I'm from the old school where even a break was considered not very good if you are in the university. So university, you're just reading until perhaps you go be sacked. But it's good these things are changing. It's now not bothering you. Guys, just give me a few minutes. I'm taking the register. But you can talk, we can talk on other matters. You can let me know how things are going with my course. You can tell me, Obila, here, things are not going very good. Or Obila, here, things are very good. Some compliment, I also need them.
The course is not too bad.
Ah, good.
Although
It's
we did,
interesting.
yeah, like personally, I didn't know that it entailed such details.
You thought you were just coming here to scoop a A and then you walk away. I assure you, not in my class. Those guys will tell you that Ovila will just teach you like a mad person until you become doctors. So by the time you interacting with the doctors in the disease control process, you will never get scared of anything. You know, when you meet with a group who are many than you are, sometimes they tend to want to not even threaten you, but you feel threatened that, hey, will I contribute anything here? Because these guys come from different training while I did another training. It's not the case. You know your staff. When it comes to those computer gimmicks, they will not understand. But at least you have a capacity to contribute at some degree. When we talk about gram-positive of the cause disease, you know. So when they think there is an increased level of gram-positive in facilities, you know what they're talking about. That is the idea. So we crafted it with a lot of foresight into it. So don't worry. Yes.
But it's nice. It's not a bad cause.
No, it's not bad. It's not bad. By the way, you guys are very lucky that you're doing this. I think Dr. Some told you that you are among the very few in sub-Saharan Africa, if any. I don't even think in Johannesburg or Pretoria people do a lot of such work in the context of disease control. In what it's usually done at master's level, but we'd say people who have understood other courses, so they only come for a smaller swath of it. You guys are going to form a huge base. In the Europe and US, they have these courses, and which is making those individuals very, very lucrative. I think they are paid better than most of us in the outright health sector. So when you join us in the health sector, you guys get more money than myself, and I may just feel a bit, jealous. But that will be my own problem, because why didn't I do serious things? So I have how many people? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Vanessa?
Present.
Vanessa is the co-host. And then Michelle Kirimi. Michelle logged.
Present.
Michelle, I almost marked you absent that you logged and went away. Masi Mucho.
No.
Present.
Great. And then Jura was there earlier. We talked. Jura?
Present, sir.
Great. So I don't have to check with anybody else. the statistics say correctly that all of you are present. Thank you very much for coming, and we will